All right, we are going to be covering chapter 10, and this is on DNA, looking at the chemical nature of DNA. So this image that you see here, which is found in chapter 10 in the beginning, is a picture of a Denisova cave in Siberia. And in this cave, scientists found a 90,000 year old bones from an ancient human. Specifically, once they analyzed the bones of the human, they revealed that there were two copies of sequences located on the X chromosome. This indicated, one, that the bone came from a female. The thickness of the bone suggested that she was at least about 13 years of age at the time of her death. And they've named this female Denny. And it showed that her DNA, when they analyzed it, was a hybrid between two extinct groups of early humans, the Neanderthal woman and a Denisovan man. Okay, now the reason why I'm opening up with this, and you'll see throughout this semester, each chapter I try to open up with some type of case study or application of what the chapter is going to be about, is it allows you to understand the longevity, the importance and significance of DNA. And you'll see that this will be much significantly different from RNA. DNA is I'm going to say, it's tough. It is able to withstand time and still have value. Okay, considering this 4,000-year-old species or woman, the bones, they were able to isolate DNA from the bones and do analysis on it. So this suggests the maturity and the importance of DNA, and how it is much more sturdy, right, than that of, let's say, RNA, which is not. And we're going to go into today, hopefully, your understanding of the chemical and the physical properties of DNA and how that is to be. So let's talk about some key characteristics of genetic material. Okay, genetic material must contain complex information, instructions for the traits and functions of an organism. So that's what the complex information is referring to, the instructions for traits and functions of an organism. It must also replicate faithfully. That means the genetic instructions must be copied and passed on with fidelity. So it has to be able to be copied and passed on to offspring. The genetic material must be able to encode a phenotype. So what you see in the DNA must transcend to phenotypically what we can see with our eyes. And a mechanism for that genetic instructions in the DNA has to be copied into RNA and proteins. We'll talk about that concept of gene expression going from, DNA to RNA, which is transcription, and then RNA to protein, which is the process called translation. That genetic material must also have the capacity to vary. That concept is understood as genetic variation. Okay, but we have to understand an important concept before I continue on, and that is our understanding of genetic material has evolved over time. Okay, what do I mean? In figure 10.1, you're going to see a timeline of science and a lot of contributions of studies from scientists that really helped us understand DNA. Okay, one being Miescher. Okay, his study with nuclein and our understanding of what that is now being termed nucleic acid. Okay, where he did pus studies. He collected bandages from people in a ward that he was working in, and inside these bandages, he found this white substance that was consistently in all of the bandages of these patients, and he isolated it and coined the term nuclein. Okay, which we now know is nucleic acid. You also have Kossel, who was instrumental in understanding that or contributing to the idea that DNA contains 4 nitrogenous bases: adenine, guanine, thymine, and cytosine, or A, T, C, and G. We also have the concept of Chargaff's rule. Chargaff's rule is referring to the ratio of bases, that there is a 1-to-1 ratio of bases of purines and pyrimidines. And so this is how we were able to, as you'll see coming up, understand the complementary base pairing of DNA, with adenine binding with thymine and guanine with cytosine. Now, keep in mind, though, that this study came after Levine, okay? Phoebus Levine. Phoebus Levine studied the chemical or the chemistry of nucleic acids using the saliva of dogs. And he was known for discovering nucleotides, which was somewhat incorrect, but it laid the foundation for further studies. So the whole concept that I want to point out to you is, and this is the figure 10.1 in your textbook or ebook, it shows you this timeline of how we have science and medicine has progressed, and each experiment, each observation and series of studies really contributed or laid the foundation for the next set of studies that were to come. Here's our Miescher study. We talked about in the first set of chapters the Schleiden and Schwann cell theory, Mendel's work, and then you're going to see here's Levine, his proposal of the tetranucleotide theory, which was somewhat incorrect, but was laying the foundation for what we need to know. But then we're going to get into some more experiments like Griffith's experiment, which was very instrumental in what we know today. So, who is Miescher? Okay, he, as I mentioned earlier, was the individual that performed the study from looking at the pus of bandages in patients and coined the term or discovered what was nuclein, what he termed nuclein, which we now know was nucleic acid. This was a very instrumental study because it laid the foundation for the next series of studies that were to come. So this is an example of Chargaff's rule, and what Chargaff's rule suggests is that there is a equal ratio of adenine to thymine, guanine to cytosine. Okay, so there's an equal ratio, and you see this in various sources of DNA, whether it was E. coli, yeast, rat, human. That ratio was about a 1-to-1 ratio, and this was important because this allowed us to scientists to build off of the theory and our understanding of DNA complementary base pairing and the actual structure of DNA. Okay, so DNA is known as the source of genetic information. It was identified as the transforming principle in Griffith's experiment, which I'm going to show you in a second. But it also laid the foundation for 2 other hallmark studies that you should know. You should not finish this course without knowing these 3 hallmark experiences, especially those that are going on to grad school or another type of terminal degree program in the health sciences, or any terminal degree program. You need to know these experiments because they are really important and they do show up on the MCAT. So I want to go through some of these experiments. All right, with Griffith's experiments, his question was, can an extract from dead bacterial cells genetically transform living cells? What his experiment started off was he used virulent strains of bacteria. So he had the type 3S and type 2R. S, some may refer to it as smooth, and R for rough. And this was really referring to that polysaccharide coating on the outside. So what they found was they injected the bacteria into the mouse, and the mouse died. So when they injected the 3S, the S strain of bacteria into the mouse died, and they were able to recover the virulent strain of in the mouse. When they injected the rough strain, the 2R strain, the non-virulent strain, into the mouse lived, and they were not able to recover any bacteria. So they decided to heat kill the S strain, because the S strain, remember, when injected into the mice, died. right? And one of the reasons why, and there's various theories about this, but one of the reasons why they decided to heat kill it is because for a long time, you'll see they thought that protein was responsible for heredity, and so it is believed that that polysaccharide type coat, they thought it would also deactivate protein. So they heat killed it, and the mouse lived. So this actually gave more. It fired up more questions because they wanted to understand, okay, was it the polysaccharide coat? Was it protein that wasn't activated? So the next study, or the next part of the experiment, is they mixed the heat killed S with the R strain. So they mixed B and C together. And then they injected that into the mouse, and the mouse died. Okay. But what's interesting is what they recovered. They recovered the virulent strain in the dead mouse, right? And so their conclusion was that a substance in the heat-killed virulent strain of bacteria genetically transformed the R bacteria into live or virulent bacteria. So there was some type of transformation, and this should not be a new term. We talked about this in chapter 9. Okay, so that is the transforming principle study. Avery, McLeod, and McCarty's experiment built off of Griffith's experiment. But in this case, they did the same thing, but what they used were enzymes that specifically destroyed the components into question, because at this time they didn't know what was the transforming principle. Was DNA responsible for heredity? Was RNA responsible, or was protein responsible? So they used the same concept of the virulent strain with the R strain, but what they did is they treated each of these subsets with an RNase, which is an enzyme that destroys RNA, a protease, which destroys protein, and DNase, which destroys DNA. And what you'll see is only the culture that was treated with DNase did not undergo that transformation. Okay, and so this study allowed them to make the conclusion that DNA was the transforming principle. But scientists, we always don't just, we have questions, and we always need sure proof and evidence to support theories. So you have an experiment by Hershey and Chase where they use bacteriophages. We've learned about T4, as I mentioned it before, but you also have a T2 phage, bacteriophage. And this is a bacteriophage that infects E. coli. Here you have your phage. It has DNA in the capsid of the protein coat. Here is your tail fibers. The phage infects the host bacteria with its phage chromosome. It incorporates itself into, breaks down the bacterial chromosome, and allows for the transcription and translation, the production, of the phage particles and components that will then allow the progeny phage to reassemble and to lyse out of the bacteriophage and the bacteria cell and infect other cells. This is synonymous with what we talked about in chapter 9 of the lytic cycle. So given this information, the Hershey and Chase experiment utilized their understanding of bacteriophages to look at and to further support what was the transforming principle. So to do that, they used bacteriophages, but they used E. coli that was grown in two types of media, radioactively labeled sulfur S35 or S30 or P32. The reason why they use either sulfur or phosphorus is because the presence of either of these two are determining factors in the substance that is being transformed. And so this is why they used sulfur and phosphorus. Phosphorus is found in DNA. Sulfur is found in protein. So they grew the bacteria up in these various components, and as the bacteria is growing up, it's going to incorporate this radioactivity into the either DNA and or protein. They then allowed the phages to infect the bacteria. Then they sheared the protein coats off in the blender, separated the proteins from cells by centrifugation. And then after centrifugation, they looked for radioactivity, in the offspring. In the S35, after centrifugation, the radioactivity of S35 was recovered in the fluid containing the viral coats, but no radioactivity was detected in the progeny, the offspring. Okay, so this was an indication that potentially protein was not the transforming principle, because the radioactivity should have been identified in the progeny, and it was not. However, in the radioactively labeled P32, after centrifugation of the infected bacteria, they found P32 radioactivity in the pellet, which is where the nucleic acid, the DNA, was. Okay, so the progeny phages had radioactivity in them. This suggested that DNA, or further confirmed what the previous experiment showed, was that DNA, not protein, is the genetic material in bacteriophages. So, why is this important? Okay, all genetic material is encoded in the structure of DNA or RNA. Okay, Watson and Crick was instrumental, along with Rosalind Franklin, in helping us really understand more about the structure of DNA. And they did this by utilizing the data from Rosalind Franklin that showed what we call X-ray crystallography, or X-ray diffraction of DNA. So Watson and Crick was able to take all the history of information that I previously showed you on that timeline of various theories that were proposed and data that was proposed to look at nucleic acid and the transforming principle, such as Chargaff's rule and Griffith's experiment. But they found an interesting piece of data that was really the important component, and that was the actual X-ray crystallography data. And what happens is, with X-ray crystallography, you have crystals of a substance which are bombarded with X-rays, and they cause the light to diffract, and they create an image, okay, on a photographic film. And you'll see here that it looks like this X-like structure, which is an indication of a viral of this helical-like structure, which is DNA. And from that, they were able to determine, that DNA was a helical double helix. And combining that with the other theories that were proposed, such as Chargaff's rule, they were able to identify that DNA was a double helix, okay, that was composed of bases that were complementary base pairing with each other. So let's look at another experiment that was done to really, this was by Conrad and Singer's experiment, and they demonstrated that RNA in the tobacco mosaic virus was the carrier for genetic information. This is important to understand because, remember, not all organisms contain DNA as their primary nucleic acid. There are some that contain RNA. We talked about this example in chapter 9, where we talked about RNA viruses or retroviruses such as HIV, influenza. Those are examples of viruses that use RNA as their primary nucleic acid, not DNA. Okay, so RNA also is a form of genetic information. While most organisms do carry DNA, there are some that carry RNA, and tobacco mosaic virus is one example. Okay, and you can look at this experiment here. I strongly implore you to do so, to understand how they were able to determine that RNA using the tobacco mosaic virus was the genetic material for this specific virus. And just quickly state, tobacco mosaic virus is a virus that infects tobacco leaves. Okay, and it is a very virulent virus, and if it does infect, let's say, a farmer's tobacco plants, they actually have to burn everything and get rid of all their tools, because it is not easy to dispose of once an infection occurs. So let's look a little bit into the structure of DNA. Okay, DNA stands for, as you all should know from your previous classes, deoxyribonucleic acid. Now, if you break down that term, deoxyribonucleic acid or ribonucleotides, D meaning without, oxy referring to oxygen, ribo is the sugar or ribose. So when you look at DNA, on the ribose, the sugar, the second carbon does not contain an oxygen. That is where the deoxy comes from. Okay, on the sugar ribose, there's an oxygen that is missing here. Okay, there are three parts to DNA or a nucleotide. There's a nitrogenous base, Those are the bases that we talked about in Chargaff's rule, A, T, G, and C, or adenine, guanine, thymine, and cytosine. You also have the ribose, which is the sugar, okay? And notice the placements of the carbons on the sugar. There's 1, 2, 3, 4, and then this is the 5th carbon. That is going to become important when we start talking about 5' and 3'. And then off of the 5th carbon, you see there is a phosphate group. Now, the phosphate group does not always have to be off of the 5th carbon. You could have a phosphate group which is off of also the 3rd carbon. This is how we get an alternating sugar phosphate backbone in DNA. The bases can be classified or grouped into purines and pyrimidines. Your purines are going to be adenine and guanine. Your pyrimidines are going to be cytosine and thymine. Again, this is just showing you the diagram that I showed you before, but it's allowing you to see the difference between RNA, or the ribose in RNA, and the ribose in DNA. The ribose, or the sugar in RNA, the 2nd and the 3rd carbon is oxygenated. However, in DNA, the 2nd carbon is deoxygenated, so there is no oxygen on the 2nd carbon, and this is a clear difference between the two. I want to also further tell you that when we talk about and you start hearing about sequencing of DNA, the oxygen on the 2nd and specifically the 3rd carbon is going to be very important for chain elongation. That is replication. Okay, that oxygen is essential for chain elongation. Here are your purines and pyrimidines, as I previously mentioned. The only other pyrimidine that I did not mention yet was the uracil, and that is because uracil is the base that is present in RNA, not DNA. So in RNA, instead of having thymine, you have uracil. This is your phosphate group, okay? And this phosphate group is very essential. And one, it carries a negative charge, which makes DNA, acidic. But this phosphate group is essential in the 5th carbon atom in the nucleotide, and also bonding with the 3rd carbon. These are your images of your nucleotides, which are nucleosides with your phosphate or phosphates. So here's your nucleotide, your phosphate, your sugar, your base. All right, and here, are your bases, okay? And you'll see here when you look, there are different abbreviations for them, depending on if you're talking about a nucleotide, the actual nucleotide symbol, the nucleoside, and the nucleoside symbol. So here is a diagram of a DNA strand, and you'll notice, and also a diagram of the RNA. Notice one of the clear differences between DNA and RNA is DNA is double-stranded, RNA is single-stranded. This is one of the reasons why in the very beginning I opened up when we talked about our case study of our early human Danny, and how they were able to isolate the DNA and analyze it from over 4,000 years ago, because DNA is very stable. It is much more stable than that of RNA, and it has to do with its structure. DNA is a double helix. It is a 2-stranded molecule, and the bases are encapsulated by the sugar phosphate backbone, as opposed to RNA, which is single-stranded and does not have, it only has one sugar phosphate backbone. So RNA tends to be less stable. It is less stable and is more easily degraded than DNA. Notice here you have hydrogen bonding between the two bases. There's your thymine and your adenine. What holds together your adenine and thymine bases is hydrogen bonds. There are other electrostatic forces that contribute to this bond, but the primary bond here is hydrogen, and it holds these two bases together in a complementary base pair manner. Adenine and thymine are held together with 2 hydrogen bonds. Guanine and cytosine are held together by 3 hydrogen bonds. And you have this antiparallel complementary DNA strands. One strand is going from what we call the 5' to 3' manner. Notice that 5 and 3' is referring to the 5th and the 3rd carbon on the ribose. And on the parallel strand, or the opposite strand, it is the opposite. The ribose is inverted, and it's the 3' or the carbon, or the 3rd carbon, that is actually on the at the top, and the 5th is at the bottom. So they are anti-parallel. Here you have a diagram, again showing or a secondary structure of DNA, and this is showing the B form of DNA. There are different forms of DNA, A, B, and Z, but this is showing the B form of DNA, okay, showing it from different perspectives. The two, what we call polynucleotides, those are the two strands of DNA, they run in opposite directions, therefore they are considered anti-parallel. The B structure right here that you see, the bDNA, is a right-handed helix, which has approximately 10 bases per turn. Okay, and then you have these spaces between the backbones as it is turning. You have what we call the minor groove and the major groove. Okay, these are spaces that are you're able to see that occur between the DNA, and you'll see as we start talking about genetic mutations later on in around chapter 18 that these spaces or these grooves in DNA are actually places that allow for chemicals to intercalate and cause DNA damage. Okay, this is showing you the three forms that I was just talking about, and one of the clear differences between these three forms of DNA is the direction of their turn. bDNA and aDNA are all both right-handed turns. That means they have a clockwise spiral. However, the B form of DNA has about 10 base pairs per turn. The A, and it's more elongated. The A form, while it has the same turn direction, it is much shorter and wider than the B DNA, so it's more compacted. The Z form of DNA is left-handed, okay, so it's counterclockwise in its turn, and the sugar phosphate backbone zigzags back and forth, giving rise to its name. This is why you have where the Z DNA comes from. When researchers looked into zDNA, they found that the zDNA specific antibodies bind to regions of the DNA that are being transcribed into RNA. This suggests that zDNA may play some role in gene expression, and it also has a high GC content. All right, so this diagram here, which is about in your textbook as well, shows what we call major information pathways. So it's showing you in figure A what we call the central dogma, okay? And that is DNA is transcribed into RNA, and RNA is translated into protein. However, I want you to know that there are some misrepresentations or some exceptions to the central dogma that make it somewhat false, okay? Because it gives if you look at this pathway for A, it is unidirectional. That means it suggests all DNA is transcribed into RNA and all RNA is translated into protein, and that is not true. As science has progressed, we understand that there are instances, as we learned in chapter 9, that there are some organisms that have RNA as their primary genetic information, and they actually can go in the opposite direction and reverse back to DNA. So this is where B comes from in this terms of special information pathways. There are some viruses that are RNA based, not DNA based, so they can revert back into DNA via the process of reverse transcription. Another caveat to the central dogma that you see in pathway A is not all RNA is translated into protein, okay? As you see here, not all RNA is translated into protein. There is some RNA that does not become protein. Okay, so there are some caveats or exceptions to this pathway here. Some examples of specialized structures of DNA and RNA, they're hairpin structures, okay? Hairpin structures are very instrumental in RNA that has more of a functional role but does not become protein. For example, your tRNA is composed of hairpin structures. It does not become protein, but the ability of it to fold in such a way that it forms this hairpin-like structure is essential for its ability to help with carrying amino acids to the ribosomes for translation. We also see hairpin structures that play a part, as we'll talk about in DNA replication, in the dismantling of the apparatus when replication is occurring. Okay, so not just DNA, RNA also forms these hairpin structures, and one of the foundations that is believed to contribute to these hairpin structures or these loops are what we call palindromic sequences, and these are sequences that are the same but they're read in opposite direction, and so it allows for complementary base pairing to form, i.e. hydrogen bonds. Let me show you that example here. Okay, so you'll see here is that hairpin loop. Okay, this is the structure in this linear form. Once it forms that hairpin loop, you'll see here specifically, I'm going to highlight this portion right here for you. You'll notice here that that sequence is the same read in opposite direction, but notice complementary base pairing occurs, that hydrogen bond between A and T and G and C. So this allows for these hairpin-like structures to form, giving this dimensional shape or structure to DNA and RNA. Some other important factors that I want to point out to you in terms of the structure of DNA and RNA is methylation. DNA methylation is a process by which DNA can be methylated by adding methyl groups to predominantly cytosine and guanine. Okay, so these methyl groups are added to nucleotide bases, and it's believed that these bases, once that are methylated, have a direct correlation between gene expression in eukaryotic cells. Okay, through a process that we call epigenetics. Okay, and this is where genes can be silenced or not expressed, or they can be overexpressed just by the addition or the removal of methyl groups. If you don't remember what a methyl group is, it is a carbon and 3 hydrogen molecules. So CH3 is a methyl group, and adding a methyl group to let's say cytosine at the 5th carbon makes it 5-methylcytosine. And methylating cytosine can cause the blocking of the apparatus to access the DNA to prevent proper transcription or replication, I'm sorry. Okay. Methylation also is an essential part in bacteria and how old versus new DNA is identified as well. So the addition of these methyl groups is essential. This is chapter 10.